Now we'll move on to look at the brain. The internal structure of the brain, which is extremely complex, lies outside the scope of this atlas. In this section, we'll look at the main external features of the brain and also at the cavities within it, the ventricles. This model shows the shape of the ventricular system. It's formed by two small cavities in the midline, the third ventricle and the fourth ventricle, and two much larger cavities, the lateral ventricles, which connect to the third ventricle here. It's the third ventricle because the lateral ventricles are counted as the first two. The ventricles are filled with cerebrospinal fluid. We'll see more of them as we go along. To understand the external features of the brain, we'll start with the central stalk, which is known as the brainstem. To look at it, we'll take the rest of the brain out of the picture. Here's the brainstem. It consists of the medulla, the pons, and the midbrain. The brainstem contains tracts that connect the cerebrum, the cerebellum, and the spinal cord. And it contains nuclei that serve basic autonomic functions. It's also the origin of nearly all the cranial nerves. The medulla is cone-shaped. It tapers down to become continuous with the spinal cord. The medulla becomes continuous with the spinal cord here at the foramen magnum. The medulla, the pons, and the midbrain are located just behind the basilar part of the occipital bone and the dorsum celli. The dorsal aspect of the medulla faces almost directly backwards. The back of the upper part of the medulla forms the floor of the fourth ventricle. On the model, this is the fourth ventricle, this is the floor. This arch of tissue is the superior medullary velum, which forms the roof of this part of the ventricle. This delicate tissue, the inferior medullary velum, forms this part of the roof. This cut surface is the attachment of the cerebellum. It's described as consisting of the superior, middle, and inferior cerebellar peduncles, which are somewhat fused together. The ventral aspect of the medulla is marked on each side by these bulges, the pyramid and the olive. Emerging from the ventral and lateral surfaces of the medulla are the filaments of the four lowest cranial nerves, the 12th, the hypoglossal, the cranial part of the 11th, the accessory, the 10th, the vagus, and the 9th, the glossopharyngeal. Here's the brainstem in situ, seen from behind. The tentorium has been removed to give us this view. Here's the cerebellum, divided in the midline. Here's the divided cerebellar peduncle. Here are the filaments of the hypoglossal nerve, making their exit from the cranium. Here are the accessory, vagus, and glossopharyngeal nerves, making their exit together through one opening. Above the medulla is the pons. On each side, the pons becomes continuous with the middle cerebellar peduncle. Arising from the groove between the pons and the medulla are the next three cranial nerves. They're the 8th, the vestibulocochlear, the 7th, the facial, and the 6th, just visible, the abducent. The 5th cranial nerve, the trigeminal, emerges from the upper part of the pons. Here's the middle cranial fossa, here's the petrous temporal bone, here's the pons. Here are the facial and vestibulocochlear nerves together. Here's the trigeminal nerve, here's the abducent nerve. Each cerebral hemisphere contains a cavity the lateral ventricle that's filled with cerebrospinal fluid. The lateral ventricle has an anterior horn, a body, a posterior horn, and an inferior horn. The anterior horn is in the frontal lobe, the body is in the parietal lobe, the posterior horn is in the occipital lobe, and the inferior horn curls downward and forward into the temporal lobe. To see where the lateral ventricle communicates with the third ventricle, we'll go round to a medial view. The communication is here, at the interventricular foramen. To see how the lateral ventricle, the third ventricle, and the fourth ventricle are connected, we'll look at a brain that's been divided in the midline. Here's a midline section through the third ventricle. Here's the third ventricle. This strand of vascular tissue in the roof of the ventricle is the choroid plexus, which produces cerebrospinal fluid. Here's the interventricular foramen opening into the lateral ventricle. The choroid plexus passes through the foramen and continues into the lateral ventricle. The cerebrospinal fluid that's formed in the lateral and third ventricles passes through this narrow passage, the cerebral aqueduct, and into the fourth ventricle. Fluid leaves the fourth ventricle through three openings: the lateral apertures, the right one is in the depths of this recess, and the medial aperture, which is in the midline, here. It's easier to visualize the medial opening in this dissection. Here it is, in the inferior medullary velum. The lateral openings are here. The medial opening comes out here, between the cerebellum and the medulla. The lateral opening on each side comes out just below the cerebellar peduncles. These openings lead to the subarachnoid space surrounding the brain and spinal cord. We'll see where the cerebrospinal fluid is absorbed later in this tape when we look at the blood vessels. We'll return now to the underside of the cerebrum to look at the structures that form the floor of the third ventricle, which is here. Here's the optic chiasm. Behind it, this tubular structure that's been divided is the infundibulum, the stalk of the pituitary gland. These two projections are the mammillary bodies. To see how the pituitary gland is attached to the brain, we'll look at an intact specimen divided in the midline. Here's the floor of the third ventricle. Here's the optic chiasm. Here's the infundibulum, leading down to the pituitary gland or hypophysis. The anterior and posterior parts of the pituitary gland are quite distinct. The pituitary gland sits in the pituitary fossa. The pituitary fossa bulges downwards into the roof of the sphenoid sinus. This area just above the pituitary stalk is the hypothalamus. In the dissections we've seen so far, including this one, the picture has been simplified by removing the important arteries that surround the base of the brain and run in the major sulci. We'll see these later in this tape.